Now over to the panelists. Can you present yourself? From the right, we have only one academic, but he is, of course, a tiger. Come on, Don.
Thanks, Bent. I'm Lon Schneider. I'm from the University of Southern California and work in a couple of Alzheimer's disease centers that have worked in translational research for a while. I was absolutely staggered a few moments ago when Roger said that we were going to do this as though it's a World Economic Forum panel in Davos, because I immediately thought and realized that I left my skis at home, and where are the parties? You know, the parties, the champagne and the food. So now I'm disappointed that I came here and did not get to ski, I think, so.
Okay. Word to Hank.
I'm Hank Saferstein. I'm with Cognition Therapeutics. We're a discovery and development CNS company with a clinical stage asset, CT1812, in phase 1b studies. We have a first-in-class small molecule that effectively displaces A-beta oligomers from neuronal binding sites and clears them into the CSF. The CT1812 was derived from our high-content phenotypic screening platform and unbiased, biased chemical platform. So really pleased to be here with a distinguished group of entrepreneurs and clinicians, and looking forward to the discussion.
I'm David Elmole from the Harvard Medical School, and also I'm the founder and chairman of AZ Therapies. The company is in a phase 3 trial all over the world, and we already screened over 500 subjects with many randomized, hoping to have an interim analysis by the beginning of 2018. And I just would like to say in the beginning that the ocean of approaches, scientific approaches, is huge, and actually we cannot say which one is going to be very successful. It's just the trials will show it is successful. The second part is all the scientific approaches actually work, at least in the beginning or in part of them. And then maybe some toxicity causes the trial failure, or the trial design because of heterogeneity of the study, the population of the subjects, and all kind of other things like the diagnosis, how the trial was run. So it's not just one simple thing that can cause the trial failure. Also, Alzheimer's disease is not really cancer. In cancer, you treat for maybe 3-6 months, and then all the side effects, when somebody is really have a remission, starts going back, and he is and become normal again. In Alzheimer's, you really have to treat for 1, 2, 3, 5, 8 years, and that's why the administration of chronic toxicity really makes a lot of sense, and causes a lot of failure. So hopefully, in the way we are doing it, we are doing it early. We are treating 2. It's a combination therapy, probably the first one in the field. We are treating the 2 main pillars of the Alzheimer's progression, and I'm mentioning progression because that's the most part that we need. The goal is really not to cure Alzheimer's, but really to slow down or prevent the progression of the disease. So, and we are doing it by 2 ways. One is to try to stop the neurotoxicity from the oligomerization and polymerization, just by putting a molecule that stays in between the 2 A-beta that all of us are producing, and letting them wash out with time. And the other is to try to deal with the invisible neuroinflammation in the beginning of the disease by treating it to stop the neurodegeneration associated with that. So these are the two things that we are trying to use, and hopefully we will be successful.
Good afternoon, everybody. My name is Martin Tolar. I'm with a company called Alzion. We're a Boston-based biotech, and we're fully focused on Alzheimer's disease. We founded the company about 5 years ago, and at that time there were 10 years of failures, and now there are almost 15 years. So I think this panel is very appropriate. There are a number of targets, but as you know, hundreds and hundreds of these studies and programs have failed. Our insight has been that the disease is very heterogeneous, that we need to find a population that actually we can test the drugs in. We're drug developers. And we really decided to take the playbook from cancer and apply the precision medicine for Alzheimer's. And what it meant that we wanted to find a population of people that clearly have the disease. We can easily diagnose them. We obviously have a genetic risk, so we can focus on the genetic diagnosis. We know that they have overexpression of the pathology. And if the drug works, that targets this pathology, we can then expand to the whole population. Our insight has been to focus on ApoE4 risk factor, which, as you know, has been uncovered almost 20 years ago now, but became more and more in prominence because it is now the second most important risk factor after age, really, in the sporadic Alzheimer's. And we set out really to look for a drug that would work in the population. Initially, we focused on the carriers, but then on really homozygous population. And one of my previous jobs was running the business development advisor, so I've seen all the programs in sort of the nascent stages 10 years ago. And we searched through a number of these programs that had clinical data. We were really focused to have in-license or build a program that has already clinical data. And we came across a program for a molecule that's called tremiprosate. So it's a molecule that has been through about 2,000 patient program. The study was negative in the overall population. But we have found that actually in this very targeted ApoE4 carrier and ApoE4 homozygous population, we have found very compelling efficacy, over 5 points at 18 months time point on the cognition, on ADAS-Cog, and about over 1 point on the functional scale. So that really gave us confidence to go into confirmatory study. All of these analyses have been done obviously post-hoc, long after the programs have completed. And we developed an improved formulation because one of the drug development problem with the original molecule was it wasn't very well absorbed. So we developed a prodrug formulation that improves absorption, improves pharmacokinetics, and also is safer. The only risk factor or the only adverse factor was the little bit of nausea and vomiting. And we have completed the full bridging program with this prodrug. We also met with the FDA and got a green light for a confirmatory study, a confirmatory program in the ApoE4 homozygous patients, which we're going to start this year. I'll also mention that we were a really group of drug developers and focused on Alzheimer's. We worked over 50 programs, and this team has been together for a decade now. Our first company was a company called Comentis. We were the first ones to crack the beta secretase. We were the first one in the clinic, and did a large deal with Astellas then. Our chairman is JP Garnier, who was the former CEO of GSK and also chairman of Actelion, which has been quite successful company and started actually very similar as we did. They started in a garage, we started in my kitchen. We also, because of the insight into molecular mechanism of this drug, how it works, which appears to stabilize the native state of proteins and prevents the aggregation of the proteins, we also attracted Stanley Prusiner, who is the Nobel laureate who discovered prions, because he has been focused for the past decade on the development of therapeutics for prion diseases and amyloid diseases. And really, we're working together using our platform, our insights into the molecular mechanism to develop other therapeutics. But our key program is the one focused on Alzheimer's disease, and we're kicking off the confirmatory program for ApoE4 homozygotes this year.
I'm Christopher I'm Christopher Missling, president of Anavex Life Sciences. It's a biotech company which is NASDAQ listed. And we took it a different approach. We took a look at it, and I think the first thing I want to say, and I'm sure the entire panel agrees, and probably everybody in this room, that Alzheimer's is a very complex disease. And what we did, we looked at other indications, and when you look at, for example, a oncology space, and what is the most successful drug development in this indication right now, you know that it is immunostimulation. So when you ask yourself, what's the difference of immunostimulation and the classical targeted cancer approach, is that you're utilizing your own body. And we were asking the question, can you translate that approach into CNS? And by asking this question, we found a protein which is actually we all have in common, but it's never utilized as long as the cells are in homeostasis, are in balance. However, this protein, which is called sigma-1 receptor, is utilized when they are out of balance. And the interesting thing is that this approach is not limited by definition to one indication, because all the pathologies have in common to some extent a protein out of balance or homeostasis dysfunction. And what is interesting when we developed this approach, and the good news is that because the sigma-1 receptor is a GC-coupled receptor, so you can activate that with small molecules. And what we're doing, we are replicating exactly what the biology does, is to activate when there is a need for that. And it's a bit like a fire truck in a firehouse. If there's no fire, you never need a fire truck. But if there is a fire, you really need the fire truck very badly. And probably when you look at indications like Alzheimer's and Parkinson and others, what they have in common, they are chronic in nature. And these chronic indications are probably causing this ability of the body to help himself to be diminished. And it's a bit like diabetes type 2, where the body is not able to keep up with the need for the insulin-producing cells. But the good news is that when you activate the sigma-1 with external sigma-1 agonist, then you're able to show an improvement in the following phenotypes. We saw reduction in A-beta, we saw reduction in tau, we saw reduction in mitochondrial dysfunction, we saw reduction in inflammation, and we saw an increase in the calcium imbalance. And when you look around in the posters in the last few days, I saw a lot of posters about exactly those subjects. And that makes me, in a way, very excited, because that shows something is going on with that approach. The other thing I want to point out, that by definition of this approach, it cannot be limited to one indication. And while we have the most advanced data in Alzheimer's patients in a small phase 2A study so far, we also have now seen that the same approach with the same molecule is working in a Parkinson pathology, in a 6-OHDA model, in also Rett syndrome, neurodevelopmental diseases, but also in depression and anxiety. And we also saw other indications which we'll continue to work on. This is basically the approach we're taking, and maybe that is helpful to discuss this during this panel as a potential, addition to approach this complex disease.
Thank you. Teshi?
So I'm
Please introduce yourself again first.
Okay. I'm Teji Kimura, Chief Discovery Officer, Neurology Group in Eisai. We are now developing two amyloid-based programs. One is the BACE inhibitor, E2609 in phase 3, and the second is the amyloid protofibril selective antibody, BAN2401 in phase 2. We are still believe that the amyloid hypothesis is most promising, although there are so many negative data so far. But these data indicated the importance of the clinical design, especially the selection, right dose, right patient population, and also the right clinical scale. So, and currently that the most of the clinical program based on the amyloid hypothesis going to the very early stage, such as the preclinical prodromal. In that case, we also need to consider less invasive diagnosis method to widen the opportunity. Of course, currently we can select the early patient population by utilizing the PET scan and also the CNS measurement. But the blood diagnosis is quite important for getting more wider patient population. And also that already that Pamela mentioned, AD is a very complex disease. So just one hypothesis not treat everything. So combination treatment is a very useful like the oncology area. Even in that case, so we need that clear biomarker diagnosis method underlying that clear the rationale to determine patient population and also the intervention timing and also the treatment regimen by compound and combination by combination. So today I would like to hear a lot of the information about these points. Thank you very much.
Thank you very much, and we are really impressed how many questions we have already got. I'm sure we can answer only part of them. Ezio, do you want to start with one? Of course, we are happy if you can give us thoughts about translation, how we can bring basic research into the clinical work. But
right, I would like to remind the audience that we are talking about translation. Now, translation is about somebody who comes to Tokyo to sell whiskey. And unfortunately, he cannot translate because he doesn't speak Japanese. However, he meets another girl, an American girl who is there to sell something else. They are getting together and they have a wonderful weekend. This is a movie which is called Lost in Translation. And this is to tell you that very often we are lost in translation. I wonder of our guests here, how many are they lost in translation? But certainly, being in drug company, they know what translation is. So please ask questions which are related to the difficulty to move from animal experiment into the clinic. What is the problem? Why do we have so few drugs, new drugs for Alzheimer? What is the problem with translation?
I have really some translation question on my desk here that seems to work now. The first one is really, we'll see. Is preclinical efficacy needed for a program to move into the clinic? Do we need preclinical research to be able to move up into the patients?
I can just give an example of what we have done. First, you have a hypothesis. You want to do a combination, so you have to test both drugs and look at them in vitro if they do what you want them to do. Like if one to try to see that, for example, from series of drugs that we tried, you get one that does inhibit the polymerization and oligomerization in nanomolar amount. That's very important. Also the size, how it is working. And then you go and try it in the animals. And you can go and then try it in animal model of I would call Alzheimer, but the animals don't develop Alzheimer like in human, but it's for plaque, and you see if it really inhibits that. So after you see that it inhibits that, and you test the other drug for looking at the neuroinflammation, how it changes the microglia economy, and if it does all those in the animal model, and it could be many studies. It's not only one. You also test the function, looking at swim tests, etc. And then you put an IND after you prepare the drug for treatment, which is really also difficult and costly to try to prepare it for taking it into the clinic. So if you do those, and actually we were lucky, we did it within less than a year and a half, and we also, most of the studies were done at Harvard a long time ago, but that's the way it goes. You just go by steps, and you try to push it.
Yeah, I think this is a good question. Can we hear, Lone, your idea, being a clinician? And we heard yesterday that still all what we heard about animal proof here wasn't really working well. Can we leave away some of the preclinical experiments and move quicker into patients? How do you see on this?
So the question I heard you ask is, can we dismiss some of the preclinical work and move quickly into patients?
Correct.
And I think one way of approaching translation, at least T2 translation, is to consider whether, in fact, enough preclinical work was done to justify taking a molecular entity and bringing it into patients to begin with. And just to be provocative for a moment, I just suggest in general that much preclinical work that we rely on to bring a drug to humans is not well developed, is based on perhaps small animal studies and methodologically limited studies, and often studies that are not replicated. So if we're discussing barriers to translation, this is one barrier that's often not dealt with. We just sort of go around it based on ideas and enthusiasm. And I'll just stop there.
I would say, if I can add to this, unless there are validated models in pluripotent cell assays, for example, which translate into human efficacy, I think it's a question of safety and risk. You still have to do animal models for risk reasons. You cannot toxicology studies, you cannot avoid it. It's mandatory. And the second thing is risk. Do you want to go right into humans independently of the discussion about risk, of safety? It's a question of more expenses. So you immediately basically put yourself up to a much higher expense level and as a higher risk.
Martin, we have certainly seen some severe side effects in some of the trials directed against A-beta. Did you want to comment on this?
Yeah, I'll just answer the question about preclinical translation. So we don't have a preclinical model for Alzheimer's. We just don't. What we do, and we should be doing, and we should actually rely on those data. If we select a pathology, like amyloid, and there's an amyloid depositing mouse or some species, we should see efficacy, and that's what we call target engagement. But I think we should, and actually I think all of the speakers here have said the same thing, we should move very quickly to the only appropriate population, which is patients. And that's where I would argue we have been failing, and that's where, again, the understanding of the heterogeneity of population, how we actually select those. I mean, there were even at this meeting, there were so many new insights, actually, how distinct populations are actually in what we call the clinical Alzheimer's disease. And again, this has been the rationale for us to say you have to apply precision medicine. You have to be very clear how you define the population where you're going to test the efficacy. Because if you have 20 different factors playing into the overall population, you just can never win. And this is frankly where cancer has been successful. The reason for success of cancer is actually they did apply precision medicine, they genetically selected cancers, they tested them very specifically with targeted therapies, and they were successful. And I think that's what we need to apply in Alzheimer's. And frankly, the failures, the recent failures, the big trials targeting disease modification have not controlled for the populations. I would argue that has been the problem for those.
So I just wanted to add one thing. We give a lot of weight at Cognition Therapeutics to the value of translational models, particularly some of the transgenic mouse models. And the important thing is that, we heard this morning the power of spatial working memory, something we can test in rodents, and we can also test, obviously, in humans. I think some of the problem with the translational studies that have been done is, A, many of them haven't been published, or they just simply haven't been done, so we don't have a lot to build on. And B, a lot of the work in the clinic was done preclinically about 10 years ago. So back in the day, people wanted to move plaque. Maybe that's not so important today. Maybe we needed more behavioral efficacy in these translational models before we moved into the clinic. So there is that delay. Certainly no one's arguing here that we need to move quickly, but there is a lot of value in these translational, in these animal models. We just have to understand what to measure, and as Martin mentions, understand some of the endophenotypes that we see in patients and how they might relate to these animal models. All very difficult, but doable.
We probably can cure a lot of animals, but not the...
I have one speaker here.
Okay.
Could you replace some of the animal data in preclinical work with working on human material, or have you some other ideas of...
Okay. So, of course, that we are trying to fish to the non-human primate for the confirmed efficacy. But non-human is not human. So just that data provide biomarker change. So that is the limitation. And if we would like to translate that effect to human, we have to utilize the genetic mutation data in human. It's a more promising approach, I think.
We are moving to the next.
Yeah, thank you. We got a very good question. It comes from a very expert anonymous who I know, and the question is very good. And this is dealing with the complication of translation. This is a translation which is complicated. The question is, if we are dealing with a heterogeneous disease like Alzheimer, I suppose by heterogeneous it means we have many possible causes, I beta, tau, etc. What is the rationale of targeting, he says, amyloid for all of these variants? Is that complicated? What kind of translation? How can we translate something which is so complicated as an heterogeneous disease like Alzheimer? Please tell me how you do.
I'll comment briefly. From our perspective, all the biochemical and genetic roads lead to amyloid at some aspect of the disease. I don't think anyone here would argue that it's going to end up being a polypharmacological approach, and different stages of the disease require different therapeutics or combinations of therapeutics to treat them. And of course, again, we're going to have to understand the heterogeneity. Alzheimer's is probably a constellation of a number of different diseases. But amyloid is going to always play a part, I believe, in the equation. And the question will be, when do we treat these patients? Is there enough neuronal or synaptic real estate to rescue if that's something you're going after? And in the early stages, are there ways of turning off the tap, if you will, or preventing amyloid from taking on its toxic form and exerting its effects that way? So yeah, most of the translational models, animal models, have been focused on amyloid for many, many years, and we're starting to see people breaking away from that. Now there's tau models and other models, but I think most people here in the panel would agree that amyloid is playing a part in this disease, and it's really a question of when and how.
So I'll answer the amyloid question. This is the big question in the room. Why are we targeting amyloid? What we want to see as drug developers is something that's really hardwired to pathology, right? So we know that from the family Alzheimer's, every single family Alzheimer's disease will actually be caused by overproduction of amyloid, and it's hardwired. If you have a mutation, you know exactly when you're going to get the disease, and it's predictive, actually, that you're going to get the disease. So that's what validates a target for a drug developer. Second, as you all know, Down syndrome is a trisomy 21. You have 3 copies of the precursor protein for amyloid, or the gene for the precursor protein for amyloid, and all the Down syndrome patients will have in the teens amyloidosis in the brain, and in the 40s will have developed the amyloid, the Alzheimer's pathology. So that's the reason why everybody's targeting amyloid. The problem is, and that we have been targeting GRONC amyloids, which we all understand now quite well as well. Everybody was looking at the fibrillar amyloid, at the plaques. It's probably just scar in the brain. What we do understand, I think there are now literally thousands of papers actually, which I was surprised by because this is a fairly novel idea, is the focusing on oligomers. There is clearly a species of amyloid that's clearly toxic, that's clearly correlated to pathology, that's clearly correlated to the clinical symptomatology, and that's really the target that we should go after. Now, our approach has been really focused on this oligomer aggregation. We have a molecule that prevents the aggregation of monomeric amyloid to these oligomeric assemblies. And that gives us confidence to actually even take it forward. But I think the wrong question is, should we focus amyloid? The correct question is, which form of amyloid, and how we assure that actually we really, if we target this kind of species, the oligomeric species, it's actually we have effect in the animals, we have effect in the humans, and so forth. There's clearly interplay of many other factors. Tau inflammation is clearly has a role, but we don't understand it. I mean, we spent 20 years beating on the amyloid door. I started in this field, 20 years ago with Lon. I mean, and we knew that amyloid has to do something with it. But I think finally, at least for the amyloid axis, and because of the hundreds and hundreds of programs we have run, this is the right target. That's the reason everybody's asking us, well, everybody's failing with these huge multi-billion-dollar programs. All the pharmacists are still doing this. Why? This is the reason. And it's the right reason. We will see an amyloid targeting therapeutic in the next few years.
I want to really agree with what you just said, because A-beta is no doubt is important pathology. The question is, though, I want to take it to another level, is not only the form, but also the location of where the A-beta is showing up in terms of its toxicology. We just have, and it will be presented at this conference in 2 days, that in the endoplasmic reticulum, the A-beta oligomers are doing damage and interfering with the mitochondria. So that's something to be said about the A-beta. But you just also mentioned, Martin, about the effect of other pathologies like inflammation and tau. So another question, go back again, so why A-beta, right? So that's kind of like comes back to the question which we initiated it with. But I think the fact is just the complexity is so high that the only thing we know is A-beta because it is a low-hanging fruit. And it's obvious to go after that first. So there's nothing wrong with trying that approach. But the next thing should be to take it a notch higher. And as Rudy Tanzi also noticed, the complexity is beyond A-beta being toxic is also sometimes even protective. So what is it now? Is it bad? Is it good? So we don't know. So the way I think we should maybe think about it, let's maybe the body decide where and what to do with A-beta. And this is a little bit, I come back to the approach of immunostimulation. Let the body decide if the beta level is too high, it will take it down. If it's at the right level, it's protective, it might not touch it.
Yeah, we have another question that is really directed to you, David. It's a personal question from an anonymous. They ask, what is published data that ibuprofen cromolyn hits their target in aging human brain? And what studies supported dose finding for a phase 3 trial? So we are over in the inflammation, infection.
It's a very nice question that is directed actually to our trial. We have done quite many studies in animals, at least 3 groups of 75, and not by us, but CRO. We looked at the change in the A-beta amyloid at different doses in the animals, the changes of A-beta, all the mesoscale, A-beta for 38, 40, and 42. We also tested the behavior of the microglia in the economy of the microglia in vivo. And also we looked at the plaque accumulation at the end of the study. Plus, we have done also a swim, the Morris maze, to look at the function Okay, so all those were studied in the animals, and we determined from calculations how can we titrate the amount of A beta that is produced in the brain every day, and not try to give huge amount a dose that really can cause side effects and toxicity. So that's the approach that we have done. We calculated, when we have done human studies that look at the pharmacokinetics in blood and in the CSF. And we can see how much, what's the amount of that we have in the blood and the CSF with time. One of the things that we saw, that our drug actually washes out for more over than 8 hours from the brain. So we know that the dose that we used is the one that being used in human. And we do only one dose per day.
Okay, thanks. It was a question only to you. We have a question directed more to you, Lone. It's a clinical question, but still about the future. It's must discussed if we should use a more cocktail form of treatment, a combination treatment. And the question is, what combination therapies are we most likely to see in the clinic first? BACE inhibitors and anti-A beta, or anti-inflammatory in combination with anti-tau, as two examples.
I only get a choice of two. A.
Choose any combination.
A or B. You know, talking about combination therapy is interesting from a number of points of view. One is that the patients we're treating are heterogeneous and are defined by mostly clinical characteristics with one or two biomarkers supplementing that. And the next part of the combination treatment would be, as you're asking, what combinations would you use? I think it's particularly formidable to actually answer that question. We're not doing well by identifying one drug and treating a group of patients, and we're not seeing any effect. We're not seeing proof of concept with virtually any individual drug. So I turn the question around and say that the first thing you'd have to do, we would have to do, is be able to identify our population based on a phenotype, and then based on the observation that the targeted that we're or the identification of two phenotypes that we are then proposing to treat with two particular drugs, so that it's not an automatic kind of syllogistic logic. Oh, Alzheimer's disease is characterized by amyloid and tau. Therefore, the amyloid is bad, the tau is bad. Let's target amyloid, let's target tau, and we'll get a better response than one or the other when we've been unsuccessful with a single drug. So I'm avoiding the question entirely.
So I would agree with Lon. The problem we have, quite frankly, for 15 years, that nothing works. Nothing. We are not able to get a positive signal. And the first challenge is going to be, again, with a single drug, find a population or find some sort of a group of patients where we can see efficacy. There's clearly going to be combination therapy. I'm confident of that. But we have been failing for 15 years. So for the drug developers in the room, I mean, it's even more complicated, quite frankly, to get approved something as a combination therapy, especially if you have no idea how the individuals work, right? So theoretically, it looks very nice, but practically, we really need to find the right trial design, which actually we should probably talk about. You know, what are the insights actually we have learned recently, going early, you know, selecting the right population and so forth, but we need to have a win. I mean, you all appreciate how incredibly depressed, you know, the field is, because for 15 years, everything has been failing. I'll give an example. When we were about 3 months old company, still in my kitchen, David Cameron, the British Prime Minister, organized G8 Summit. on Alzheimer's. He invited all the world leaders and say, what are we going to do? How we're actually going to address this disease? And a lot of the companies didn't show up at all because they have abandoned Alzheimer's, like Pfizer. You know, Pfizer dissolved about 500 people group just about at the time. And really, the problem again, this Alzheimer's is, as we all know, it's not only the biggest problem in medicine, but it's one of the biggest problems facing society. We would have to rethink the end-of-life decisions. We would have to rethink how we deal with actually so many people, because no society can deal with tens of millions of people, but we need a win. And the problem is for anybody in drug development, even in the pharmas, is that we have been failing and nobody's willing to take the risk.
I do think
We
that
have now heard two rather pessimistic views. Now I want more optimism from you both.
Sure.
Please. Otherwise, you are quiet.
I'm a little bit more optimist, but to add on what they already said, that there is a lot of other things that have to be done. You have to look at the interaction of the two drugs and how they affect the genetic toxicity and others. So we have to do that. And plus, I have about probably 90 patents that are protecting all kinds of combinations that we don't know which one. You just have to take the ones that maybe are going to be the easiest to translate from animal studies into human. And that's what we did.
So I think we can take a page from the oncology playbook. I mean, we've seen 20 years ago, actually 15 years ago, combination therapeutic development simply on biomarkers, where we had two experimental therapeutics that were acting on different aspects of the disease or different therapeutic points of entry. And those studies have now blossomed into bona fide combination therapies that are in labels. And I think we should be pursuing the same thing in Alzheimer's disease. You know, for instance, monoclonal antibodies, not a lot of that gets into the brain. We know that. So is there a way to displace targets and then clear them with monoclonal antibodies? We can start to think about combining anti-inflammatory therapies potentially later in the disease. But what's important, and what Lon mentioned, is that we have to understand how to segment these patients. And in the context of combination therapy, that really comes down to biomarkers. Well-defined biomarkers that will help you understand if you pull this lever, you can move these biomarkers in this direction, and this lever will move a few other biomarkers, or there's some synergy between two therapeutics on a set of biomarkers. So that's what we need to be thinking about. I think that's what we're all doing now, trying to expand the knowledge base around molecular biomarkers so we can more accurately address the various kinds of patients that we're seeing that have Alzheimer's disease.
No one has mentioned the possibility to combine a pharmacological agent with non-pharmacological. I mean, we know a lot more from epidemiology about risk and protective factors. We know about environmental factors. Is that something you are thinking about?
But we're in the same position with environmental interventions as we are with drugs. We still don't know the target or the essential component. You can easily enough show, for example, that in any given group of people, whether they have Alzheimer's disease or not, that physical exercise and aerobic metabolism will be associated with some improvements in cognition and some increase in hippocampal volume. But you still don't have a mechanism and you don't have a dose response or an essential therapeutic quantity to that. So yes, it's fine to blend, let's say, an exercise intervention with a drug intervention, but you have the same issues in trying to decide who the target is, what the intermediate outcomes should be, and what the therapeutic potential of the combination is. So,
Are there more comments on this question?
Okay.
Otherwise, we move on. Have you a new?
Yes. I like to bring back to translation because I like the word translation. I received 3 questions which I will summarize because they are all about the same. Yesterday we had a 1-hour discussion about models, animal models. And this comes back again about which is a good animal models. All of you people have been working with animal models. And one of the questions is, mice are cured of all diseases in studies, but often does not work in clinical trial. Shall we look at using different animals in preclinical studies? I don't want you to elaborate too much on what you think, but what did you lose the faith, or you still have a faith on animal studies? And what is the problem? Why so many showed efficacy then it was not efficacious in human?
Because we don't have a model for Alzheimer's.
We don't have a model.
We don't have an animal model for Alzheimer's. Period. We have animal models for pathologies, and we should test those. We should have a clear target engagement. We should affect on the pathology. If a drug doesn't do what you expect it to do, it's not working. It's not actually ethical to take it to humans. But we need to go to patients. We need to go quickly to patients, and we need to test it in the patients.
But we can go straight to patient.
Well, the basic drug development process where you have to test for toxicity, where you have to say for bioavailability, all the basic has to be done in animals. I'm not questioning that at all. But we're focusing just on the efficacy, because that's where we're failing. We have drugs that get where we want it, they do what we want them to do, but they don't work. And again, the problem is in the patient populations, in how we run the studies, how we design the studies, and what patients we enroll in those studies, period. And again, that has been the lesson over the past, at least a decade, and we know how to do it now. We have learned tremendous amount from all the failures. We call it at Pfizer clinical discovery. That's the only way you can actually learn. But you cannot go to the animals because it just doesn't translate.
Okay.
To your
Dr. Kimura, your company had quite a experience. You were pioneer in developing animal models. Actually, you were the first to develop the good animal model. Would you tell us what happens to animal models now,
Yes.
20 years later?
Okay, thank you. So, yeah, and I think that the primary endpoint in clinical trial and readout animal, it's that one of the issue. So, of course, that we can look at biomarker change, either the animal and clinical human. But that if we consider some cognitive function, it is a completely different in animal and human. So that is the one big question. So we really need a more objective readout even in the clinical. So for example, the EEG or some MRI, functional MRI, if that such kind of the objective readout become the primary endpoint, maybe that the translational is much easier than the currently one. That is my comment.
Thank you very much. Other opinion? Yes,
Yeah.
please.
One thing in the field of imaging, you can go even to the monkeys and take all the monkeys, and some of them really develop the Alzheimer. But to try to deal with doing an animal study in monkeys, first of all, it's going to be more expensive than human, and also the measures for the test that you are doing, the batteries of tests that you are doing for cognition and function, and also to look at the biomarkers in the human, it's much easier going through the animal model. So you don't, like I heard before, you don't have a choice. You can look at the targets, answer some of the questions, and look at the toxicity, and then go into human studies and try it.
Yes, Lord.
Can I suggest something, maybe to try to get to your point? It's we can engineer a drug against an animal model target. So, as an example, aducanumab is a great example of an antibody well engineered to do what it was intended to do, knock down amyloid plaques. It does that in animals reliably. It does that in humans. However, in the process of engineering a given drug to a target, to an animal target, is not the least predictive of whether there's going to be a clinical response in the selected human target. So the next step I'd suggest is that if you have a well-engineered drug, whether it's donepezil or aducanumab or others, you would expect it to have efficacy in a group of people who have the target or who have the deficit. We'd expect to see that in a relatively small number of patients, like less than 400 with donepezil, reliable effects. And we'd expect to see with aducanumab reliable clinical effects in a relatively small number of patients. Specific criticism: it should not take 1,250 patients in 2 studies in order to hope to see an effect with a well-engineered drug. And that would then extend to any of the drugs we advance into humans. We should be able to identify the patients and be able to see that the drug shows target engagement and shows a clinical response fairly quickly. And if it doesn't, dump the drug.
Everybody agrees with Dr. Schneider?
It's a terrific comment,
Yes.
and I think probably we have to help the FDA start looking into changing a little bit the direction and not having a huge trial. Our trial is lower from the point of view of number of subjects, but we lose quite a bit because of the way we are trying to homogenize the group that we would like to test. So that's really cost us quite a bit in the screening process because there are many failures, screen failures, because we try to have this small group that have the same thing and progress the same thing.
So just to build on what Lon said, there's no disagreement that there are drugs out there that target the target very effectively. The question is, does targeting that target actually lead to some benefit, cognitive benefit or otherwise? And I think we've learned over the years that while plaque is problematic, it probably is not the toxic culprit that's leading to this widespread synaptic regression. So, I think it gets down to really understanding, what exactly is your drug engaging, what target, and how to link that target to some kind of behavioral outcome that you could reach in a reasonable size study. And this is not what we've seen in the past. Hopefully we'll see more of this in the future. Drugs that are more efficient or selective at getting at the toxic culprit and therefore driving down the number of patients that are required to demonstrate efficacy.
So if I can...
Just very quickly, we have not demonstrated that plaque busting is not effective. We just haven't done the studies yet to see whether it is or isn't. So I'm not sure that we're ready to move past that or that we all agree. We just haven't done the studies. We may be doing them, but haven't done them.
So as you said, should it be smaller trials? I fully agree. It should be, right? It's driven, the powering for the studies is driven by 2 things, by a signal. And we have been, and there were a lot of recent examples where we focused at very, very, very small signal. We should not run these kind of studies because they, frankly, even if they show statistical efficacy, statistical significance, they will not help patients. And second is the variability. So we really need to have, again, we need to have a drug that clearly engages the target and have a population where this pathology is overexpressed, which is the only way you can actually have a smaller studies. I mean, our powering, we will have about 200 subjects per group in our pivotal study, so we don't need 500 or 1,000 patients per group because it's, again, driven by variability, which we decrease with the precision medicine approach, focusing on the ApoE4 homozygotes. And second is the signal, where we have a strong enough signal which warrants actually this kind of confirmation. I can agree. And this is the reason why, again, the value of a treatment for Alzheimer's, this is going to be the largest medication. Wall Street says this is going to be $100 billion market. So that's why the pharmas are beating on these tiny signals with these huge studies. But it's not the way to do drug development. I fully agree with you.
But can we reach a consensus here that despite all of this discussion, the conclusion is that we have no choice, we need animal models?
We need animal models.
You are
Of course,
not
we
in agreement
need the
that we
right
need
animal models.
animal models?
We need the right animal models.
Do we have animal models?
We need the right animal models. But I think I want to add to what Lon says, and I understand his frustration. The frustration we all share is that why do we have to have a failure in a phase 3 study? It shouldn't be a 2,000 or 1,000 phase 3 study failure. A phase 3 study should be to confirm the efficacy for the drug. And I think Professor Windblatt did a phenomenal publication where he pointed out that the dilemma is that the moving from the preclinical into the phase 3 is just happening too quickly in Alzheimer's. Is that correct? And this is exactly the dilemma. So a proper proof of concept study is the reason why we see this huge patient's failure, and that is frustrating. So maybe it's better to go back and look at what has also been done in oncology, population pharmacokinetic. So very much looking at the very detailed, it's more hard to execute, but it's important to do that right away because it addresses target engagement, it addresses drug response curve in multiple endpoints, including EEG biomarkers and A-beta biomarkers. And then, only then you should say, if this is all matching up, then you should go into a phase 3 study. And I think that might have not happened yet.
I'm coming to a more ethical question. It's from Alison. Alison is saying, so much focus is now on early treatment. Do we ignore those living with frank AD? Can we also focus on improved cognition, for example, in contrast to disease modification? I mean, could we see effects on behavioral and psychiatric symptoms and function and cognition and not really focus on the pre-stages of dementia? Perhaps even some studies on more severe stages of the disease.
I must say that the goal that we had when we started to try to look at what we would like to do is actually to try to change the present statistics that one of every 3 of us here, by the time that we are 80, will be a subject of Alzheimer's disease.
No, 1 out of 4.
Oh, okay, it's even worse. No, actually, I'm sorry. But to change it to 1 out of 4 would be with Alzheimer's disease by the time we are 90. However, we should not really forget about the ones that already Alzheimer's people and try to find something to improve the quality of life. And that's really a different type of behavior. It's probably to increase the firing of ligands in the brain, doing some other things that really can improve the normal behavior of a person. And it's and that could be maybe spread for 1 year, 2 years, and not for really modifying the disease.
I'd say that the focus on going earlier and earlier really has 2 flavors. The first is, the mentality of pharma and small companies not wanting to let go of an asset. So it didn't work in mild to moderate, maybe prodromal. So unfortunately, there's that, right? Maybe it needs to go in earlier. There's some indication that some therapeutic strategies would work earlier. But one of the things that I don't buy in on at all is that by the time you present with dementia, in mild to moderate, if you will, that the game's over and it's a endless cascade of horribles that end in vegetation. I think that as long as there's synaptic real estate, that you can protect it and you can bring about some decent quality of life, no matter what the stage is, and we need to be focused on that as much as we are on prevention and disease modification.
I will respectfully disagree. There is a congruence of evidence from clinical studies, from pathogenic studies, and so forth, that you really have to intervene early. And the drugs actually, at least the drugs targeting amyloid, actually do have effect in the earlier stages. Our drug has shown exactly the same thing. And really, we're at this stage, while we all neurologists here, most of us are neurologist clinicians, we would love to help all the patients. We just need to have the first win. We have been failing for 15 years. We need to find a population where, at least with the target we understand the most, which is the amyloid, where we have the first efficacious drug or an efficacious study. And I think, again, at least for the amyloid targeting, it's very clear. You have to go early.
I would just again say that.
I would agree with both of you. So it's the irony, right? So I agree. First of all, we need a treatment right away. So you have symptomatic, clinical situation. You have to have something for a patient. So we have the responsibility to develop something right away. But also we have to find something for MCI. But even I go one step further. We need something for prevention. So what about we find something which everybody can take in the morning, like a small aspirin or like a Lipitor? This is the way to go. And I agree, obviously, that there's been a more pragmatic reason for going earlier, but there's also indeed a pathological reason for that. So I do agree for both arguments.
I must say I don't agree completely with you. My feeling is even if we come in early, we perhaps keep the patients on a somewhat higher level for a longer time. But let's say they even live longer, and the final stages are still in the severe stage. I mean, that stage might be the same number of years or even longer. So I mean, we have to see that the problem with the population is also a caring problem. It's very much a community problem. The cost is in the community. The small cost is on drugs and on assessment. So I think we should look on every possibility. I like the question.
I fully agree with you, man. But I have the drug developer hat on, and our goal is to find a treatment that would show efficacy, and the FDA would actually approve, so we can give it to patients. And while I cannot agree with you more, I take care of a lot of patients, and it's tragical, at every stage. But we need to find something that works. And again, we have been failing for 15 years. And nothing showed, we have never been able as drug developers. This is drug development problem, where we actually, I think there's a slide at the end of the tunnel. I'm convinced we're going to have a treatment targeting amyloid within the next few years. But this has been the problem. We have not understood the disease well. We didn't understand heterogeneity. We didn't understand the specific populations. We didn't understand what to target with the drug, and so forth. We have been learning, but again, we're coming to the end of the tunnel, and we're clearly going to have effective treatments in the next few years.
Well, I'm perhaps more looking on treatments like affecting the serotonergic system, MAO-B inhibition, glycine, more of these classical studies that still improved the severe stages of the disease. And these are absolutely not done today.
Right, and I fully agree. So there are two ways to treat Alzheimer's: symptomatic, which I cannot agree with you more. The drug development on the aggression, and the psychiatric symptoms, on cognition and so forth is going to go forward. But the challenge for us as drug developers, researchers, and frankly humanity, is to find a drug that would prevent the symptoms, right? Ultimately, as somebody said here, you want to have a drug that you can actually give patients who are at risk for the disease. I like one of the studies that Biogen has done. They show that 28% or 27% of 65-year-olds have amyloid in the brain, and they probably will progress. These are the patients you want to target. 16 million people just in the US. So we have 5 million patients with the disease, 16 million they are on that path. It's very interesting that it's 28% because probably these are the ApoE4 carriers. But again, this is your population. So we will end up in the future in the same situation as this cardiovascular disease, where we're going to treat amyloid, at least that one understanding, amyloid the way we treat cholesterol. We will look at, do you have a risk factor, how old you are, and what sort of information we need. But again, we have to win at least once to be able to do that.
So,
Okay. Always we have to consider that the economical benefit by the drug treatment. For example, effect size just at the 25%. Maybe I don't think that not so much that the economical benefit. But if the effect size 75% slowdown of the normal AD, cognitive deficient, maybe that patient can live that normal condition. So maybe that by end of their life. So in that case, that the economical condition completely good. So we always consider how much effect size is necessary, not only the drug effect, but also the financial condition, I think.
Okay.
Can I get in this? Because I think that this interaction here is an example of the semantic quagmire we're in talking about things like disease modification, or are we treating early or late, or why are we focusing on one area or another. So before I knew I was going to sit on this panel, I was given an assignment to write, to give a talk on something else, on essentially drug development, and then Avram Fisher said I couldn't do that. I had to sit on the panel. So I tweeted out 12 slides of what I would have spoken about, and you could go look those up if you want. But when the question came up before, disease modification versus symptomatic, let me just address it by saying that this is a distinction without a difference. And it's largely an invention of industry and some of my colleagues that has a marketing valence to it, but really and practically is not, are not differences. And the idea of disease modification gets conflated with the need in some cases to treat long term. When the question comes up, why are we not treating, or are we treating people who are more severe or more moderate or more mild? And then, Ben, when you answered it as well, it should be a question of fairness. We should remember to treat people who are moderate. This is kind of putting the cart before the horse. I think we should be treating or targeting the people who we think will better respond to the drug that we have at hand. And when you say we want to go earlier, we want to target. Well, anyway, let me stop, and I think I made enough points for this moment.
Okay. I have the question of the questions. And the question is, tau is the key. And I will allow me to elaborate just for a second.
Tau is the key.
Tau is the key. So they want us to discuss tau. But I would like to translate the question this way. There are now 7 clinical trial phase 1 on tau immunization. My question is it easier to translate for tau that it was difficult to translate for beta amyloid? Is tau more translatable? Think not only about the clinical model, but the animal model, but also think what tau does and what are the symptoms related to tau. Is it easier to translate?
What we know about tau is that it correlates with the Braak score and the dementia severity very, very well. I attended today the Biogen presentation, and there's a very well described from Chris Rowe of the entire spectrum of diagnostic in the space. And what he said very clearly was tau is right now the least precise to be measured. So the concern would be that the precision of the diagnostic and the correlation with the drug effect might not be very good as it is with A-beta today. The other thing is that I think there's also publications out there which again make this not trivial, that in certain cases tau is actually also not always bad. And there are also different species of taus, which makes it even more complicated. So in a way, I have to come back to the previous notion of let's the body decide if he wants to reduce tau or keep it or change the level. The good news is about what we can say about our compounds. They do reduce tau. They reduce tau and GSK3 beta. And for that reason, not directly, by the way, but indirectly because of the stimulation of the sigma-1 receptor, which restores homeostasis. So we believe the restoration of the homeostasis will allow the cells to do the right thing, what is best for the body.
I think we all agree that tau is a good target. The problem is that we just don't understand the pathways as well as we do with amyloid. I think tau is about 10 years behind our understanding of amyloid, and we're just going to have to see. We're going to learn a lot from the studies, but they are very early, and we again will probably have a lot of failures and an understanding of that whole pathway. The question is again, what is going to be the first drug that's going to be effective?
I think tau is just one of these ocean of targets that one can look at. And it's just as important as VM, other amyloidosis, and like other targets. The biggest problem is with the tau is you have a taurex that already fell. And the molecule is about 10 times the size of the A-beta. So it's not as, and has 3 different repeats in normal and 4 repeats in abnormal, and then you have how do you differentiate between them and how do you treat one or not the other. So it's not as an easy question as it sounds, but one should also invest in trying to look at the tau as one of the mechanisms that may affect the disease, and how can we do it. Tau is also part of the tabular system. It's really inside, intraneuron, and it stabilizes the tabular system, that actually it's the wiring system. So before the neurons start to degenerate, this tau doesn't fail because it doesn't phosphorylate, because it has a lot of activity to do. And I think it's probably an event that happens later. And I think Dr. Tanzi showed it in at least his 3D in vitro methods, that the tau comes later.
Wasn't it a nice paper recently in Alzheimer's Research and Therapy where they treated mice with antibodies to tau and also removed the amyloid? I think it was by Khaled Iqbal. I mean, it was really a new thought that we perhaps, and vice versa might be true too. But
yeah,
mice models are mice models, that's a problem, of course.
But when you already have amyloids in the brain, I think neurons are dying. And if they...
Not absolutely.
Not absolutely, but some of them.
Some people have a lot of amyloid.
As we speak. So.
All the time. So I think it's really, it depends on how significant is the damage to the brain at that stage. And that's where the early and late comes into play. If you have a real estate of the neurons there, maybe if a treatment is going to be useful, and it doesn't matter if it's a monoclonal antibody, biologics, a drug, whatever it is.
Yeah, and also that tau mouse model should mimic that propagation pathway like the human. Just that the overexpression does not mimic human.
But I have a very specific question for you as a clinician. Tau, as we all agree, is closely related to cognition. So my question is, will it be easier to translate the effect of any anti-tau drug than it has been with A-beta, which is disconnected from cognition? Will it be easier or it will be the same?
Well, I think it'll be just about as hard, except that we have the potential advantage of advances in technology to help at this point. We already see with anti-tau drugs that we're making the same mistakes we are making with anti-A-beta drugs and others, either advancing alleged anti-tau drugs, anti-aggregators into humans far too quickly without showing the least bit of target engagement, or alternatively, when you think about the clinical expression of tauopathies, thinking, oh, we have a tau antibody. What should we do with it? Oh, let's give it to patients with tauopathies. Well, what patients have tauopathies? Oh, supranuclear palsy. Okay, let's test our tau drug in progressive supranuclear palsy. And, we're restricting our ability then to better develop the drugs. But where tau might become interesting is in our ability to recognize non-amyloid and other other causes of cognitive impairment and tau-related cognitive impairment, and then our ability to perhaps use new tau ligands to, in effect, stereotactically or spatially identify the distribution of tau in individual patients. We have the potential ability to roughly Braak stage living people, and in that way perhaps target them appropriately with an anti-tau agent along a set of stages. Now we're kind of doing what cancer, we have the potential to do the cancer clinical trial analogy, or the set of basket trials or umbrella trials by combining a tau ligand with an anti-tau or an anti-tau drug, targeting it using, let's say, tau PET.
So what
Thank
Lon
you.
described, what we call in drug development, is confidence in rationale. So the confidence in rationale, the tau clearly is a pathology in the brain, but the confidence in rationale for drug developers is much, much lower. Because again, we don't understand the pathway, we don't understand the targets, we don't understand the population as well, and therefore the risk is much higher actually in any of these approaches.
I have a more personal question to you, Ezio, and to all of the members of the panel. We are speaking about translational research in drug discovery, and here you are sitting representing small companies, big academia, big pharmaceutical company. But have we optimized the environment? I mean, my experience from working in Stockholm, we had AstraZeneca in a city 50 kilometers south of us. We never interacted. We have the preclinical drug discovery, target finding research, excellent, but how much do they collaborate with the clinic? Shouldn't we also completely reorganize the environment to have an effective drug discovery? I mean, bring patients together and basic scientists and drug discovery people into one building. What is your thoughts, Ezio? You have.
I cannot agree more that this has actually been disjointed, and this is one of the big problems actually we had in our strive to find a treatment. And we really should have the way Nixon declared the war on cancer. We should have a war on Alzheimer's. Again, David Cameron organized G8 Summit, all the world leaders, the most developed economies. What are we going to do about Alzheimer's? There were a lot of efforts. We met in London, several years after that. Nothing has happened. I mean, I think really, again, this is one of the biggest problems we're facing as a society, as humanity, to find a way to deal with this disease. I mean, it's going to be uncontrollable. It's not, in the US, it's estimated the cost is going to be $1 trillion in a few decades. If you look at our GNP in the US, you see what proportion is. We cannot survive that. Even the US, even the most developed economies. That's why the world leaders did only G8 Summit on the disease, which is on dementia. Nevertheless, nothing really came out of it. There was an effort, to get organized and so forth, but there's no concerted effort. There's always, strive to go to Congress to put more money on Alzheimer's. Alzheimer's is the most underfunded, actually, important disease at an NIH level, right? I mean, it's just not happening. And I really kudos to the pharmas that's still doing it, Eisai, Biogen, Roche, Merck, despite all the failures, Lilly. But I think this is a problem that is very well suited to biotech, actually, where you have really very focused, very concerted effort, where you actually, and trying to unlock the one problem, which is finding the right study, right population, and the right drug target, and actually test it out. And this is what actually the U.S. Biotech can do, and actually this is what U.S. biotech has been very effective in. And I think this is where the hope is. Again, kudos to the big pharmas, but I'm really pleased that there are so many biotechnology companies here actually willing to do that. People were laughing at us when we started. They call us the intrepid soldiers of Alcyon. They were like, "You are insane." The big companies, actually, the big pharmas, Pfizer's of this world, running away. How are you going to do it? Because actually we focus on the small problem. We're not going to do the big problem. We're not going to do huge studies. We cannot do that. But we can focus on the small problem and have a very targeted approach. And that's really, I think the biotech can have a huge contribution to this. But again, I cannot agree, Ben, that it's 15 years overdue. to have actually this concerted effort.
I
between
think
all of
it's
them.
really a great question, really. And I think the Crick Institute in London has now 1,500 scientists that are working in all different areas of neuroscience, neurodegeneration, and they talk to each other and they present to each other. So it's very important. It happens only when the government starts getting involved, and all their monies that they get is from government mostly. So I think until the government gets involved, it will be very hard to get that environmental, but it's really needed. It is needed.
I have to answer to my colleague. I think that to translate, you need several skills. You know that because as a company, when you are recruiting somebody, for instance, at higher position, you want a translator. And you know this translator has to know a little bit about chemistry, pharmacology, biology, etc. In addition, the change which has happened recently, where there has been a dislocation of knowledge from the big company into a vast number with less, perhaps, close collaboration with universities. And I believe one of the aspects that you are thinking about, that the enormous knowledge that is in the laboratories, in the university, has to benefit reciprocally. So it has to be used more. So the environment is very important, and the environment has changed enormously. This morning I was talking with a representative of a large company, and they say now Mr. Trump will cut 20% of the NIH budget. What will happen? It will happen that there will be less research, there will be less young people getting into research. Now, what about, are we, big drug company, have to take back all of this? So they have, this is a fear which I think is premature, but in any case, who is going to do what? I think both have roles. So the environment is enormously important. And the collaboration between, and this is the case of Sweden, which has an enormous reservoir of neuroscience, and I'm not sure that all of them is exploited to the benefit of translation. So translation is a difficult operation.
So I'll follow on that thought. So I think the sharing of the information is absolutely essential, has not been there. And this is the premier Alzheimer's conference in the world. And I think there is a time for a message to the Alzheimer's community and actually to the drug development community and say, let's share, right? Again, I worked at a big pharma. Big pharmas usually don't like to share, but actually let's really share all the data so we can learn from these mistakes and actually have a better chance at it. I cannot agree more. Again, the time has been 10 years ago to do that. I mean, and I think with preeminent leaders in the field, if you actually send that message, it would resonate. People do hear it. Again, the world leaders get together to figure out how to deal with Alzheimer's. But they're not going to solve it. They knew, actually, David Cameron, we were sitting at a table, tiny table, and he said, how can I bring you back? I said, you have to fund it. You have to actually put money behind it because it's incredibly risky. The Wall Street will not support this because they see that everything has failed for 15 years. Big pharma say, if I can invest in some orphan disease, I know that the confidence in rationale and my risk is much, much lower. Why would I should go into Alzheimer's? I'll wait. And that has been what really destroyed effort and the field. So again, time is now actually. I mean, we should send a message and say, look, we have to collaborate, we have to share the data, we have to be very transparent, and this is the forum to do it.
Right.
So Martin, you're getting a letter from me asking to share the original transit-primate clinical trials data.
Say it one more time.
I said I'm going to send you a letter asking if you'll share the original transit-primate clinical trials.
Yeah, I mean,
data.
we publishing,
Okay.
right? We published our data in the Journal of Prevention of Alzheimer's Disease last year.
Share the data, though, not publish the data. Share.
Yeah, absolutely.
Share data.
I mean, we're willing to disclose, and we have been disclosing our data to. I mean, we don't have a problem really with this kind of confidentiality because, again, I mean, we want actually people, we want researchers, we want actually other pharmas, we want actually other collaborators to really rip it apart. So we have been doing it actually for the past 5 weeks. We said, we're missing something. Is it actually for real? And I would be willing to do it definitely. We have no problem actually with the IP with this or so forth, because the IP is still around the molecule. So the data should be actually freely available.
Okay, by the way, what's David Cameron doing these days?
Well, he's not fighting for Alzheimer's, that's what I know.
I just make a comment about the alignment of resources. I think it really comes down to the funding sources we've all discussed. And I've seen models where there's a philanthropic or foundational funding source for early stage and some of the translational work, proof of concept, if you will, and preclinical models, and even some early clinical work. And then there's more of a venture-based fund approach for clinical development. And in that way, investors or donors understand what they're getting into and getting the returns, whether societal or monetary. I think the G8 summit did lead to some work. I remember there was a $100+ million fund. I don't know whether that actually was a result of the G8 summit, but the UK did invest in this fund that's being managed by a venture firm, looking at early stage research in particular. So I think the alignment of resources, whether it's academic or for profit, really comes down to how you structure the financing of those efforts. And if we do it smart, then I think we'll have the right handoff between the high-risk philanthropic approach, where donors are really looking for multiple shots on goal, to a more focused venture-based return on a specific set of assets that are being developed for a specific market.
Yeah, I think really that we all agree then that we need more collaboration between academic sites, clinical sites, and industry. And for me, co-localization would be the optimal way, because when you meet at coffee, you understand the patient's problems, and clinicians can give comments on the preclinical part, and certainly in between having the drug discovery people. That would be an optimal, quick way into the patient, and I think that we should work on that. So thanks a lot. I understand the IP problems, and I think we have to be, coming from the academic side, I think we sometimes feel the companies are not generous enough to us. They keep all the IP rights, even if we have been in the discovery. But I think coming together, we will understand each one has difficulties, each one has economic problems, but collaboration is the key word for translation too. Are we closing now? Yeah. Should we have a round of comments about the future? What is our vision now for drug discovery, for coming up with new drugs? How will this knowledge about translation help us? Could we start from this side now?
So, Alzheimer's disease is still a very severe disease, and there is no disease modifier so far. So I think that at least we have to provide a disease modifier to the patient as soon as possible, even though that the effect size is not enough. But that is the starting point for the disease modification. I already mentioned, so maybe that the combination with other MOA and enhancement of the current amyloid-based drug is the second step to increase the effect size. But now we should concentrate how to provide that disease modification as soon as possible. Thank you very much.
So I made the rounds at the conference, and I was extremely encouraged by what I saw, that most scientific posters and presentations were really pointing out the pathology is more complex than thought. And obviously, it's about time, obviously, right, to figure that out, to acknowledge that. And maybe that also is giving ways to appreciate the complexity and, again, maybe try to learn from what has been successful in oncology, like with Opdivo, immunostimulation in cancer, and we can translate that into CNS, and maybe that is a new novel approach we can think of.
we have learned over the 15 years from all these failures, and I would argue we're at the tipping point. And I would argue that my prediction is over the next few years we will see an effective drug that's going to get approved. Most likely it's going to be amyloid targeting, but again, we should try all of these, and we really should work together to accelerate this because the patients are waiting, and any year, any month we delay it, it's a huge problem for everybody.
I must make a really kind of comment. The Genome Project actually shows that there are human brains that had ample amount of tau and tau tangles and beta amyloids, but they had total cognition and function before they died. And the reason is, one of the reasons that they think that are trying now to look at is really looking into the neuroinflammation. That may be the one that makes everything to develop the disease. So we didn't talk here about the neuroinflammation, but I think it's very important to know that the change in the microglia and how it protects, how it toxifies the brain and protect it is the next stage of looking at the neuroinflammation, etc. But from the point of view of having drugs, I believe all the drugs are equally, in that ocean of drugs, are equally starting from the beautiful science. We just don't know where it's going to go.
So first of all, this has been a great panel. I appreciate the opportunity to be here. We've had a lot of discussion about the failures in the past, and clearly they have been failures, but I think I would argue that these failures have been due actually to poor clinical tools and poor clinical trial design. That's not a negative comment on the bold folks who are taking the risks and developing these drugs. It's just, I think, a reflection of the desperation in this space. I think we have a better handle on that now. We're segmenting patients better. We're understanding mechanism of action and target engagement much better before we leap into these large and long clinical studies. So I think the future is bright. It's going to be a challenge. Financing is always going to be a challenge. But we have to, I think, remember that we're progressing this field sort of along the lines that we saw with oncology 20 years ago, and I have every expectation that we'll have the same outcome. That was once intractable disease will be manageable, due really, again, to the bold and courageous efforts of small companies and large companies. So again, appreciate the opportunity, and we also did make some slides for this talk that are on our website at cogrx.com, so you're welcome to go see them as well.
Thanks. So I came into this intending to make a number of points, and David started it out, and that is, there, in thinking about neurodegeneration in general and so-called Alzheimer's-related pathology in general, just so clearly there are too many targets to deal with one at a time. And you can see this and appreciate this on any path map, on any in any exercise in sequencing. It also stands to reason that if you interfere with any individual target, it's likely that there will be a compensation or a reaction that will nullify what you've just done, unless that target is at a particularly important point in a cascade. So then, as David pointed out, Alzheimer's disease is not amyloid, and it's not a positive PET scan. And a positive PET scan will take, amyloid PET scan will take you just so far. In fact, in general, most people will with a positive PET scan will never have to deal with substantial cognitive impairment or Alzheimer's disease, and will die happily with a head full of amyloid. Hope I'm that lucky. And So we have too many targets, and then in applying any individual drug to the target, we have too many ways of diagnosing patients or putting them into categories, and many of them, even as though we speak of the category as valid, for instance, preclinical Alzheimer's disease, because we have a positive amyloid PET scan, still need to be validated and still need to be understood. So too many drugs, too many targets. And then we have the troublesome phase 1 and phase 2 studies when we finally get a drug that's safe and that hits a target that's safe enough to use in humans. And that is the phase 1b studies and the phase 2a studies become not a test, not a proof of concept, but a rite of passage. You just have to do them. It doesn't seem that we need them to be positive because somebody finds something statistically significant in that and advances it to phase 3 where we're often disappointed. So the phase 2 studies, by their design and by their analysis, create exaggerated effects that then do nothing but disappoint in phase 3. And then we are shocked. We look at the Lilly solanezumab studies and we say, oh, 2 phase 3 studies and we almost got there. It's close. The p-value was 0.12. We will definitely hit on the 3rd phase 3 study when we increase the sample size to 2,300. Well, it's absolutely wrong. You could have calculated the positive predictive value of those 2 studies and realized that there's probably better things to do with that. So this, these several points are what I wanted to make about the issues in translation. I don't think it's a matter of being optimistic or pessimistic about the future. We're doing science, and we're doing as best as one can do, as groups of people can do, and we're either going to succeed or not, or come out somewhere in between. And their enthusiasm should be towards doing the science and the clinical work. So, thanks.
Thanks, Sloan. I really agree with you, Lone. And it's still, I mean, a meeting like this couldn't happen in 30, 40 years ago. And I remember when I moved in beginning of 1980s from up the Arctic Circle in Sweden down to Stockholm. I was a geriatrician. As doctors, we were very nihilistic. We had no idea about treatment strategies. We didn't know anything about the pathogenesis, why the nerve cells were dying too early in the disease. We had wards with 40 patients, closed doors in the more severe stages. And if I look what has happened now, then came the cholinergic drugs, then came memantine. Now we have a lot of ideas from research about basic mechanisms. We have seen a fantastic development about the care of dementia, from these wards now to smaller wards, specific units for day and night support for demented, day care, night care perhaps also. And so development is going on, and I think this discussion here about mostly basic science and translational ideas, and hearing your views, 15 years perhaps we have a more active treatment. I think we are looking forward to the coming 10, 15 years in a very positive way. Thanks to and you see the conference like this. I mean, it's fantastic. So many people focusing on research. So thanks to the organizers sitting in the front here for inviting us for this panel. Thanks to all the panel members, and thanks to you, Ezio, being such a grand old man in this field. You should know he is also trained in Sweden. He was professor in Stockholm. So.